Secondary databases. Firstly, let us study about secondary nucleotide sequence repository. Most of the secondary databases are the subcollection of sequences retrieved from the primary databases like GenBank or EMBL. Some of the secondary databases are just like primary database, but with the addition of annotation, software, presentation of the information and cross-references. Some other secondary databases do not contain any sequences, but the information gathered from different databases. Let us study some of the examples for the secondary nucleotide sequence database. The Berkeley Drosophila Genome Project or BDGP or Flybase is a consortium that sequenced the entire genome of the fruit fly Drosophila melanogaster to a high degree of completeness and quality. The Berkeley Drosophila Genome Project or BDGP Gene Disruption Project generates single P element insertion strains of those mutated unique genomic open reading frames. Such strains strongly facilitate further genetic and molecular studies of the disrupted loci, but it has remained unclear if P elements can be used to mutate all Drosophila genes. ACE-DB is another database which has originally developed for the C. elegans or nematode worm genome project. This repository contains the sequence, genetic map and also the phenotypic information about Caenorhabditis elegans. The very flexible schema notation of ACE-DB allows the user to modify and extend the data relationship very easily by simply editing the model flies. The Institute for Genomic Research or TIGR at www.tigr.org contains all the focus on one organism. It contains the sequence and annotation of the completed genomes, associated information about the organism such as taxon and gram stain pattern, the structure and composition of their DNA molecules and the information about the protein sequence derived from that DNA molecule. From October 2006, TIGR has become a multidisciplinary genomic focused organization with a Center for Advancement of Genomics or TCAG, the Craig Venter Science Foundation, the Joint Technology Center and the Institute for Biological Energy Alternatives IBEA and is known as the J. Craig Venter Institute. Genomes of mitochondria and other such organelles are available at the Organelle Genome Database developed by University of Montreal, Canada and is known as GoBase. From the website gobase.bcm.umontreal.ca. Secondary Amino Acid Sequence Repository or GPCRGB. GPCRDB is a database focused on transmembrane protein which is used by the cells to communicate with the outside world. This database contains multiple sequence alignment, ligand and ligand binding data, 3D models, mutation data, literature reference, disease patterns, cell lines, protocols, vectors, etc. The GPCRDB is designed to act as a data storage medium as well as a tool to aid biomedical scientists with answering questions by offering a single point of access to many types of data that are integrated and visualized in a user-friendly way. Cluster. The cluster is a database, offers an automatic classification of the entries in SwissProt and TR-EMBL database into groups of related proteins. The protein families are clustered on the basis of or pairwise comparisons between protein sequences. The clustering is based on the pairwise comparison between protein sequences. The database is connected to Interpro, which helps to integrate the information on protein families, domains and functional sites from ProSite, PRINTS, PFAM and ProDom. It has also links to the Interpro graphical interface that allow users to see at a glance where the proteins from the cluster share particular functional sites. Cluster also provides cross-references to HSSP and PDB. COGs. COGs or cluster of orthologous groups of database is another database similar to cluster available online at www.ncbi.nlm.nih.gov/COG. This is a database generated by comparing the predicted and known proteins in all completely sequenced microbial genomes to infer sets of orthologs. Each COG consists of a group of proteins found to be orthologous across at least 3 lineages and likely corresponds to an ancient conserved domain. Recently, the eukaryotic orthologous groups or KOGs and egg NOG have been added to the COG database, which provides an extended analysis of more genomes including eukaryotes. The main COG's web page contains the following principal features. A list of all COGs organized by the predicted functional category. B. Separate list of COGs for each functional category and for a variety of major pathways and functional systems. C. A table of co-occurrences of genomes in COGs. D. A list of COGs organized by phyletic patterns. E. The phyletic patterns search tool. F. The COGinator program. G. A search engine to search COGs for gene names, COG numbers and arbitrary text. H. Help, which covers the principal subjects related to COGs. ProCite. ProCite is a pattern database available online at procite.xpc.org, contains protein motifs and patterns which are encoded as regular expressions. It is a method of determining the function of uncharacterized proteins translated from genomic or cDNA sequences. It consists of a database of biologically significant sites and patterns formulated with appropriate computational tools that can reliably identify the family of protein of the new sequence. The entry in ProCite is divided as two parts, that is pattern and the related text. The regular expression is placed in the same format as Swissprot with a 2-letter identifier at the beginning of each line to show the corresponding type of information. Each entry contains the reference and the link to all the protein that have the same pattern. The related text is given as a documentation file with the accession number which makes the connection to the expression data. Prints. Prints is another database for protein sequence patterns and are stored as fingerprints. Each fingerprint is a set of patterns or motifs. The entry in this database can be divided as 3 sections. First section contains the entry name, accession number, number of motifs and crosslinks to other databases. Second section is a table that contains the set of motifs that make up each fingerprint. The last section contains the actual fingerprints and is stored as multiply aligned sequences. ProDOM or Protein Domain Database. ProDOM is another database that contains homologous domains automatically generated by comparing all available protein sequences. The comparison and the clustering is performed using the program PSI BLAST. ProDOM features are being discussed, which is taken from the website documentation. This is the result obtained from a protein domain database after homology comparison between 2 or more proteins. Here you can see the most frequent protein names which have been given, and also it compares the ATP binding transporter proteins of different organisms. Here we can also see about the alignment length, the number of domains in each family, the consistency indicator and also the database comments. Let us now study about each icon which is present in the ProDomain database. The first icon is the motif representation. This motif is a graphic representation of this family. Several families have a motif representation consistent throughout the whole database. Second, we move on to the graphic representation. Graphic representation of all proteins in this domain with the decomposition in domains. The third icon, as you can see, is the list of ProDom families which are related to the current family. Related means that there are far homologies between them. This button is not drawn if the family is not related to any other one, or for the families with poor alignment or homogeneity, norm MD values low. The next icon you see is ProDom SG. If no PDB links are found for this family or its related families, and if this family satisfies several quality checks, then this family could be a good candidate for structural determination. If present, you can click the button for more information. ProDom CG. When in ProDom, use this button to access the corresponding ProDom CG family if it exists. When in ProDom CG, use it to access the corresponding ProDom family which should always exist. The next icon is MSF. To retrieve the ProDom family in MSF format. FASTA icon. This icon is used to retrieve the ProDom family in FASTA format. RIME DDDTA. This icon is used to compute a profile using PSI BLAST against this family. You should be careful to retrieve the binary file. Unfortunately, this will not work on every architecture. This functionality is still under experimental conditions. The norm D icon. The norm D value is computed for every ProDom family. If this stamp is displayed, then the norm D may be considered as high, that is greater than 0.4, meaning the alignment is of good quality. The next icon you see is predict protein. To access the predict protein server through a pre-filmed form. The next icon you see is ESP. ESP icon is used to print a high quality representation of this family. Strap. Strap is used to see the alignment and phylogenetic tree of this family based on structure. Lastly, you see the printer icon. This is used to just print the result obtained. PFAM. PFAM is another database that contains all the important collection of information available in the world for classifying proteins. The database is developed at the Wellcome Trust Sanger Institute. The aim of the database is to develop a comprehensive classification of all known protein sequences, and it is the largest collection of protein domain families. Each family is represented by multiple sequence alignments and hidden Markov models or HMMs. PFAM families contain two levels of quality, PFAM A and PFAM B. PFAM A entries are derived from the PFAM sequence database, which is built from the most recent release of UniProt KB at a given time point. Each PFAM A family consists of a curated seed alignment which contains a small set of representative members of that family, profile of the HMM's model built from the seed alignment, and an automatically generated full alignment which contains all detectable protein sequences belonging to the family as defined by profile HMM searches of primary sequence databases. PFAM B families are unannotated and of lower quality as they are generated automatically from the non-redented clusters of the latest automatic domain decomposition algorithm ADDA release. PFAM B families can be used to identify the functionally conserved regions when there are no PFAM A entries found. An example query in terms of the protein for the protein VAV human has been discussed. The same way the query can be in terms of family name, accession number, sequence ID, etc.